I was born in Chicago, and my parents traveled around a bit when I was younger, but we settled back in Chicago. I went to the university at a place called Lawrence University, which is in Wisconsin. Became interested in neuroscience largely through the professor of psychology who taught physiological psychology, a very interesting man. And I started to do a little bit of research, and when I came to go to medical school, which had been my intent, I looked for a number of medical schools, and because it was cheapest, I was going to go to the University of Illinois. And I was interviewed by a man at the University of Illinois who was the head of the Department of Physiology, and he asked me what I wanted to do, and I told him. He said, "Young man," he said, "I wouldn't go here." He said, "Go to the University of Chicago. That's the right place for you." So I did. And the University of Chicago was unusual at that time because the medical school curriculum was considered part of the graduate school curriculum, so that the medical students took the graduate course that was offered, for example, the graduate students in biochemistry. We took the course that was offered for graduate students in other departments, physiology and so on. And we had a lot of free time. I started doing research my freshman year. I did it throughout medical school. In the fourth year of medical school, I spent the whole year doing research. We had electives, and most people did clinical electives, but I did research. By early, I'd been interested in the nervous system, and I worked with a person who was interested in stress and things like that. But I became interested in memory after the early hippocampal studies came out, and I did a dissertation on the hippocampus in memory. And I discovered something that I didn't recognize, but other people recognized later, and that is that the hippocampus is not important in negatively reinforced memory. The amygdala is. And so I did a completely negative study, which got me a PhD anyway, and I'd finished the MD. And at that time, actually, it was possible to do both of them in 5 years, and which is essentially what it took me. I did a residency in neurology under very unusual circumstances. They offered me a faculty position and a training position. I was to do 6 months of training and 6 months of teaching and research. And I was a instructor in the Department of Anatomy. I had my own laboratory. They gave me startup funding, and while I was a resident in 1962, I wrote a research grant. It was funded. I wrote another one the same year. It was funded. So I had 2 NIH grants, and I was still doing residency training.
At the age of...
Well, that's...
You were very precocious.
No, I wasn't very precocious. This was at the age of 30, 31. And the initial work I did was on sensory pathways, particularly auditory pathways. And from that, for a number of reasons that were not particularly important, I became interested in visual pathways. And I had been doing also work on monoamine pathways in the brain. And at a meeting of the FASEB Society, I met Julius Axelrod, and we got to talking, and he was, at that time, was interested in the pineal. He had Dick Wertman in his lab. Dick was interested in the pineal, and Julie wanted to do, he'd done the metabolism, he'd done the metabolic pathway for melatonin, and he was interested actually in the serotonin rhythm, and he wanted to know how the central nervous system controlled the pineal gland. So he said, why don't we do some experiments? And so what we did in my lab in Chicago, I would do lesions, and we'd take the animals to his lab and kill them and take the pineal, and we'd look at the effects of lesions, which were mainly on visual pathways. and try to understand the neural control of the pineal gland. And we found that you could knock off a lot of visual pathways, and it didn't seem to do much. And so I got interested in looking for new visual pathways, and at that time, there was a new method that came out that was largely fostered by Max Cowan, but it was the method of injecting tritiated amino acids anywhere in the brain, but I did it into the eye, then it'd be taken up by the ganglion cells, would be converted into proteins. The proteins would be sent to the terminal nuclei of the retina, and it was using that technique that we found the suprachiasmatic nucleus. But it was a deliberate experiment to look for visual pathways that had not been demonstrated because we couldn't really tie down a visual pathway for this function. So having done that, I'd spent a lot of time doing lesions as a student and graduate student. So we made stereotaxic lesions, knocking out the SCN. We made knife cuts in front of it and back of it. And we had the data using adrenal corticosterone that they knocked out rhythmicity. And so that was the initial demonstration, and it was all done in a fashion of following a set of findings and just continuing to go. And from there we went on to do a variety of anatomical studies of the SCN. But that's the essential story of how we got to that set of studies.
Very logical. But you were the right person at the right time. With new methods coming up, you couldn't have done it 10 years before.
No, it's always new methods. There's an old saying in German, if I remember, Technik ist alles. I've always had two parts to my career. I do clinical neurology. I've always done clinical neurology. I specialize predominantly in movement disorders. The reason I've done that had to do with a parallel line of work that we were doing when we were doing the visual pathway work, and that was using the same techniques to look at the projections of the locus coeruleus, the raphe, look at the organization of the dopamine systems. I actually wrote the first paper that anatomically demonstrated the nigrostriatal pathway, and that was in 1971. I did work in throughout the 70s on the monoamine neuron pathways. We were the first to describe a dorsal and ventral tear in the substantia nigra, which is the anatomy of the structure. Parkinson's disease begins in the medial portion of the ventral tear and spreads from there. Continuing did that work, that kind of work through the 1980s. We continued to work on the rhythm things. Looked at a lot of different things. We, at the same time that Steve Reppert was doing it, we looked at the development of rhythmicity using the 2-deoxyglucose method. I had Shigi Shibata in the lab for a while, and we did a lot of very nice electrophysiology that he brought the technology for. Vinny Cassone was in the lab, and we did a very nice comparative study going from marsupials to primates and looking at the organization of the SCN. And we continued those things. We discovered the intergeniculate leaflet with Pat Card when he was in the lab. I did some collaborative work with Larry Morin in that time. And I'd been chair of the neurology department at Pittsburgh, at State University of Stony Brook from 1979 until 1990, and I got tired of doing that. it became more and more administrative and financial management than academic management, and I had been a consultant at Pittsburgh for the Department of Psychiatry, interestingly enough, for years, and particularly for a center for depression, and I chaired their external advisory board. And the chair of the department, who later became the dean, kept saying, "What can we do to get you to come to Pittsburgh?" And in 1990, I said, "Give me something interesting to do." So he gave me the job of running a center for neuroscience, which the primary thing at that time was to develop a comprehensive and integrated graduate program. There had been graduate programs in a couple of departments that were part neuroscience. We pulled all that all together and developed the center as an interdepartmental entity, which still exists, and that's done quite well. As I was standing around, he made me director of the Alzheimer's Center, which I did for a few years. And then in the middle 1990s, we received an invitation from a local philanthropic foundation to begin a program with Carnegie Mellon University in the neural basis of cognition. So we were given about $20 million by this foundation. There was a director from Carnegie Mellon, and I was a director from Pitt, and we developed that program, which now has 20 faculty and 25 or 30 graduate students. It has been really prospering with the new methodology and cognitive neuroscience. So I did that for a while, and then I went back to chairing the Department of Neurology at Pittsburgh because they had a crisis, and I did that for a while. In 1970, I was due for a sabbatical, and I decided it was time to get rid of all the administrative responsibilities. So I went to London to an MRC center at Hammersmith and learned how to do PET scanning. And when I got back, I closed my basic laboratory and took up clinical research in Parkinson's disease, and I've been doing that ever since.
My feel of the research you've been doing very consistently over decades, until you stopped your animal lab, as you say, was going ever deeper into the pathways related to the visual system, circadian system. And each time we say, now we know this is the pacemaker, and these are the pathways, Bob Moore comes up with a paper and says, well, there's this subventricular zone, or there's this and that. You know, you kept going into detail and doing more and more studies showing the complexity of the system.
Yeah, we spent a lot of time working on trying to parse out the functional organization of the system. And I think one of the things that it took quite a bit of time for people to cotton to was the idea that there really are functional subdivisions of the nucleus, and that these functional subdivisions have different functions. They may have overlapping functions, but they have different functions. And in the last years that I was doing the animal work, we did some studies that showed that the projections caudal to the SCN will remove the rest activity rhythm but leave the temperature rhythm totally untouched, which I think sure explains the early data when some people had difficulty with the temperature rhythm being preserved. We showed that the dorsal pathway controls the pineal, and we did some, I think, lovely retrograde work which showed that parts of the nucleus project to some areas, other parts project to other areas. There is a little bit of overlap, but the functional organization is much more complicated. And it, as you can see from the work on the transmitters and things, that VIP turns out to be much more important than one would have projected. And then we've got all these things that are being produced by the neurons that the SCN that are not standard peptides or transmitters, the prokinetosin thing, and so stuff. I mean, it's just marvelous. And then I think the story that Steve Reppert and others, and particularly Uwe Schibler, the peripheral oscillators, and looking at the SCN as a regulator of the timing of peripheral oscillators makes the most wonderful sense. The thing looks much more biological than we had been thinking about it. But everything changes. We all know that everything is more complicated than we think it is. And if you think you completely understand anything, that is the most wonderful piece of self-deception that is possible. But when in my writings and I've been working, I always have tried to state something as a idea or a conclusion that can be proven wrong. I mean, you want to have something for people to shoot at. And you want to state things in a way that people will work on it so that it will continue to increase understanding the complexity. Well, we all know that this is an enterprise that is in search of the approximation to the truth.
That's a nice way of describing science. Mentors and students, do you have any stories you want to tell us? I mean, you don't have to tell us the worst ones.
No,
Or Pitt or Ashoff, or...
I never spent much time with either Pitt or Ashoff. I'll tell you a Julius Axelrod story. I actually spent a lot of time with Julie, and we were very good friends. In many respects, I think he was almost the best scientist I ever knew, because he approached everything with an amazing simplicity. He thought of experiments in simple things, but he discovered amazingly complicated things. And he got his PhD at the age of 44. He'd been a technician for the city of New York. One day, I was working in his lab, and he had his desk in the laboratory. He didn't have an office. And he just sat at the desk, or he was up doing experiments. And that day, he was sitting at the desk, and a series of people came in to talk to him. And they'd say, "Julie, I've got this problem, and how can I approach this?" And he'd say, "Well, you do this, you do this." And then I was working right next to him, so I was listening to this all morning. We went to lunch, and I said, "Julie, that's the most amazing series of ideas I've ever heard. How do you do this?" He said, "Well, Bob," he says, "there's only one problem with this." He says, 60% of those are probably good ideas. He says, 40 of them probably really shitty. He says, "The problem is I don't know which is which."
At least it was 60-40.
Yes,
Could have been 40-60.
that's right.
Who was there in the lab at the time?
Oh, well,
Richard Wilbur or Sol Snyder?
Both of them. It was those two. It was Jacques. I'm blocking on his last name. Lewinsky from Paris. It was a pretty striking group of people, and it was a pretty amazing place. The way the work was set up, that you did experiments for 4 days, and then on Friday they wrote papers. In the first 10 years after Julie got his PhD, he published 400 papers.
You know, at NIH, and I experienced it. You just had an idea, and you found somebody in the corridors, and you went and did it.
Yeah. I think that was NIH, and I think it was true in the universities in that time. It was very easy to get money, and you didn't have to write grants over and over and over again. The things that have happened in sciences are first that the technology has continued to change over the time that that I was doing science, and we're in a point now of transition from the clone and sequence molecular biology into looking at gene regulation and at the expression of genes and the regulation of gene expression and that kind of thing. This is going to change things immensely as well. But we went through, in neuroscience, we went through a period in which neurochemistry was abysmal. Neurophysiology was the first really advanced neuroscience because single electrode work came in the 1950s and it was very powerful. And then things like Ed Everts developing single unit recording from cortex in awake behaving monkeys, which, by the way, he spent 6 years at NIH producing nothing until he developed that technology. But that was one of the things that NIH allowed you to do. But over these years, we've continued to train immense numbers of people. We've reached the point where the NIH funding is not going to ever go up again the way it has, as best I can see. And the problem is going to be for people who reach the ages of 50 or thereabouts. The technology is passing them by, and they can't get grants. And I'm concerned about what's really going to happen to these folks, because a lot of them are in situations in which the grants are paying their salaries, and I don't know about many other institutions, but ours is such that if the NIH were to quit, they'd close 90% of the research laboratories. I mean, there isn't any local funding that is going to pay for that. I entirely agree that the scientific enterprise has changed from people really exploring new ideas. It certainly happens, but I think the freedom that I had when I began wouldn't be possible now.